Hello friends, good evening. Welcome to Sam Biology class. Today we shall discuss about Agarose gel electrophoresis. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Agarose is a natural colloid extracted from certain seaweeds. It is a linear polysaccharide made up of the repeating unit agarobiotes, which consists of alternating unit of galactose and 3,6-anhydrogalactose. Agarose gel is a three-dimensional matrix consisting of helical agarose molecules in supercoiled bundles that are aggregated into a three-dimensional structure with pores and channels through which biomolecules can pass. The three-dimensional structure is held together by hydrogen bonding, because of which the structure can be disrupted by heating to the liquid state. Agarose gels have relatively large pore size and is used primarily for the separation of larger molecules whose molecular mass is greater than 200 kilodalton. It is used for the electrophoresis of both proteins and nucleic acids. Because of its large pore size, agarose gel is routinely used for the separation of nucleic acids, large proteins and protein complexes. Agarose gel is generally used at a concentration between 1% and 3%. Agarose gel electrophoresis is a method of electrophoresis used in biochemistry, molecular biology, genetics and clinical chemistry for the separation of a mixed population of macromolecules such as nucleic acids and proteins in a matrix of agarose. In this technique, agarose is used as a porous mechanical support that stabilizes the medium. Due to the presence of pores and channels like polyacrylamide, agarose gel also serves as a molecular sieve, thereby contributing to the better separation of macromolecules based on their size. The pore size of an agarose gel depends on the concentration of agarose used. Agarose forms a gel matrix with pore size that are suitable in separating DNA fragments in the range of 0.1 to 25 kilobase pairs. Generally, the pore size ranges between 50 to 200 nanometer in diameter. In most of the cases, the concentration of agarose ranges between 0.5 and 2%. The concentration of agarose used depends on the size of nucleic acid fragments to be separated. Lower is the concentration of agarose, larger the pore size, and higher the concentration of agarose, smaller is the pore size. Agarose gel electrophoresis is most commonly performed horizontally in subaqueous mode, in which the slab gel remains completely submerged within the running buffer. For making gel, dry agarose is suspended in an aqueous buffer and the suspension is then boiled until a clear solution forms. The agarose solution is then poured into casting tray to prepare gels of desired dimension. Plastic combs are placed at one end of the solution to make wells for sample loading. After few minutes, the solution solidifies and a rigid gel is formed. The slab gel is then submerged within the running buffer, placed in an electrophoretic chamber with provisions of two oppositely charged platinum electrodes at opposite ends. The orientation of the gel is fixed in such a way that the sample loading wells are toward the cathode so that during electrophoresis, negatively charged nucleic acid fragments will migrate through the gel toward the anode. Before application, the sample is mixed with a loading buffer that contains a dense compound such as sucrose, glycerol or fecal. This raises the density of the sample so that it will sink to the bottom of the well. This minimizes sample diffusion. In addition to this, the loading buffer also includes a tracking dye such as xylene cyanol or methylene blue in order to monitor the progress of electrophoresis. Two oppositely charged electrodes that are placed at both ends are connected to an external power supply through electrical leads, and then the current is switched on. Due to differences in size, nucleic acid fragments will migrate at different velocity along the gel, and as a result, they will be separated into distinct bands. The running buffer is responsible for creating a charge gradient. It also serves to cool the gel as it heats up due to the flow of electric current. During the course of electrophoresis, the tracking dye moves at a fastest speed along the gel, leaving the smallest DNA fragment behind. Electrophoresis is usually stopped when the tracking dye front has reached the opposite end of the gel. After separation, the gel is carefully taken out and is stained with a suitable reagent such as ethidium bromide. The staining dye can also be incorporated in the gel initially before solidification. Ethidium bromide interacts with DNA to form a complex which can be visualized as it shows fluorescence in UV light in a UV illuminator. Different DNA bands that are embedded at different locations in the gel can be seen as orange-red colored bands. Agarose gel electrophoresis is routinely used for the qualitative as well as quantitative characterization of nucleic acids. It is also used for the preparative purpose as DNA bands can be excised and can be purified for further studies. Due to the presence of large pore size, agarose gel electrophoresis is routinely used for the separation of nucleic acid fragments. However, it is also useful for the study of proteins where separation is size independent. For example, in case of flatbed isolatory focusing and immunoelectrophoresis. Agarose gel electrophoresis has several applications. First one is the determination of the size of DNA molecules following digestion with restriction enzymes, as in case of restriction mapping of cloned DNA. Second one is the estimation of DNA concentration by comparing the intensity of the band with the corresponding band of the size markers. Third one is the analysis of the products of polymer chain reaction. For example, in case of molecular genetic diagnosis or genetic fingerprinting. Fourth one is the separation of DNA fragments for extraction and purification. In addition to this, agarose gel electrophoresis is also useful for the separation of restricted genomic DNA prior to southern transfer or of RNA prior to northern transfer. Finally, this technique also used for the separation of proteins, for example, in screening of protein abnormalities in clinical chemistry. So friends, this is all about the principle, procedure and applications of agarose gel electrophoresis. For more updates, please subscribe my YouTube channel, Sam Biology Class. Goodbye, good night.